You're seeing my slides and seeing me and hearing me well, right?
Yes.
If you feel I haven't changed my slide or my what I'm talking about is a little bit ahead of the slides, please just unmute and tell me, because maybe there's some delay in the slides. But if that's okay, I'm going to start my presentation. Today I'm going to talk about our research on this idea that memory representations that may lead to serodependence could be distributed across the brain in more areas and stable over time. I think it is widely recognized that our perception is not a direct representation of the physical world. There are many sources of information that shape and bias the way we perceive and act upon visual objects. And one of such biases is called serodependence, the one that we are deeply interested into. Now, serodependence is a bias towards the recent past. So things you have seen in the recent past or acted upon bias how you behave for current stimulus. And here I'm going to show you how this phenomenon is usually elicited in lab experiments. So here I'm going to show you a task, a delayed reproduction task of position information on a screen. So usually the experiment goes like this. You present a target, usually around a circle, for participants, and then you remove it from sight. And then you ask participants to reproduce the location where they think the object was shown. I'm just going to shut here the window. The parakeets are too loud for me. And then what we expect is that participants are going to place the target where they think it was, and it's going to have some response error, roughly following a Gaussian distribution. And then they place where they think the target was shown. We show another target to them, remove it once again from sight. And once again, we expect some error in their responses. But this time, since they have already experienced some targets in the past, what we have seen in the past decade of research in visual perception is that these responses are biased. The response distribution error is displaced toward the previous stimulus, as if the current percept is biased toward the previous percept or action in the previous trial. And we usually call this an attractive bias, because this response is attracted toward the recent past, in the direction of what had recently being experienced. And this phenomenon is quite ubiquitous. So many research papers have been published since the seminal work by Fisher and Whitney in 2014, where they showed that perception for orientation is biased by recently reproduced orientation targets. But this has since been replicated in experiments of visual motion perception, face perception, numerosity perception, and also in visual motor tasks, like in interceptive actions. So for example, in this paper by McKean and collaborators in 2008, even predating Fisher and Whitney paper, they had participants perform a simple motion extrapolation task. So in this kind of task, participants, they see a target traveling from one side to the other of the screen on constant speed. The target gradually disappears behind an occluder, and participants, they have to press a button at the same time as they think the target would leave the occluder. And what they find here is very similar to what Fisher and Whitney have shown in their orientation perception paper. So here, for one probe target speed of 24 degrees per second, when these probe target speeds are preceded by slower speeds, participants, they tend to delay their responses a little bit. Here, positive means delayed errors with respect to the time the target actually arrived at the end of the occluder, and negative mean they were a little bit earlier. And when participants, they have their trials preceded by faster trials than the probe ones, they tend to anticipate their behavior. So here we call also an attractive bias toward the previous trial speed. So similar to what happens in visual perception. And one of the questions that has attracted most attention and that is key to understand this phenomenon is, where does this bias come from? There's a lot of debate on the literature on whether this bias comes from early sensory, perceptual, or post-perceptual decisional working memory processes. And one thing that we think that all these proposals have in common is that for there to be a bias, for the bias to take place, there should be memory. In other words, the information from the previous trial should be stored somewhere, somehow, for there to be a bias on the current behavior. But where is this memory trace stored in the brain? There is this prominent idea in the working memory literature that the same regions that are responsible for encoding and processing information about a particular task or stimulus is also able to keep information throughout a delay period in this kind of working memory tasks. For instance, if you want to keep information about motion direction, you usually have activity in an area associated with visual motor processing, human V5MT plus area. Whereas if you want to keep information about location, you have associated activity during the delay period in the posterior parietal cortex and frontal eye fields, regions that have been previously associated with location information. But here we are talking about working memory, and usually working memory is related to models of elevated activity of neurons. And we are still not sure what kind of memory mechanism is responsible for keeping information from one trial to the next and leading to serodependence. But let's assume for a second that serodependence also relies on the same, the memory trace that leads to serodependence also relies on the same regions that process the information related to the task. Where would that be? Where would that information be stored in the intertrial interval? For instance, in interceptive tasks like the one in the McKean paper we showed, many regions along the visual motor hierarchy are associated with executing this task, like area V5, posterior parietal cortex, interparietal sulcus, dorsopremotor cortex. So could it be the case that some or all of these regions also store information from one trial to the next and then ultimately lead to serodependence? So this is the hypothesis Andreas Bartels from the University of Tübingen and I, when I was there doing part of my PhD, we had. So the hypothesis that the memory trace that leads to serodependence might be stored throughout the hierarchy that participates in executing the task, and that disrupting the activity in visual motor regions would decrease such serodependence. And to test this hypothesis, we designed an experiment where participants perform a very simple interceptive or coincident timing task. Participants, they saw a target moving from left to right of the screen at constant speed with either of 5 possible speeds. And then they have to press a button at the same time as the target hits an obstacle on the other side of the screen. 500 milliseconds into the intertrial interval, we applied repetitive transcranial magnetic stimulation at 10 Hertz and 100% of resting motor threshold. And our rationale here was that if we apply TMS over regions that participate in processing information about the task, we would be able to induce noise in such areas and thus decrease serodependence. We chose 2 areas along this visual motor hierarchy. We chose to stimulate the right human V5MT+, an area associated with processing visual motion information, and left dorsal premotor cortex, responsible for planning motor actions. We also had a control stimulation region, vertex, over the head and in between both.
Sorry, can someone confirm that they also have a problem and that they don't hear Raimundo anymore?
Yeah, I also cannot hear.
Okay. I will quickly send him a message via chat. Perhaps he can still hear us. If so, Raimundo, maybe you could leave and rejoin so that we could continue the presentation.
I can hear you typing, Michael.
Okay, yes, hello.
Hello.
Good. We couldn't hear you anymore, so now we can hear you again. I don't know, maybe go back a bit because there was some...
Okay.
It froze for a while.
Okay. Can you tell me where I stopped? Was it here, a hypothesis, Andreas's picture?
Go ahead. Yeah.
Ahead, sorry.
Yeah. We heard that. Keep going. Yeah.
Yeah, now my slides are frozen. Now, okay.
Yeah.
Task. Were you here?
Yep. This we still heard. If my impression is wrong, anyone else? Yeah, I think it was right before you transitioned to this slide.
Okay. So I'll just finish the last slide to see if everything was said, and then I'll come back to this one.
Perfect.
So I don't know if you heard, but we chose these regions, dorsopmotor cortex and human V5MT plus areas associated with performing this task. Control stimulation region vertex in between hemispheres at the top of the head. We functionally localize using specific experiments using fMRI for PMD and HV5MT+. Vertex was defined using the structure MRIs, and participants also performed the task without doing any stimulation. So far, so good?
Yes, we can still hear you.
Okay, cool. So first things first, we have to show that for this task and for this particular stimulus range, we could find serial dependence. And here I'm showing you the temporal error, which is the difference between the moment participants press the button and the moment that the target arrived at the obstacle. Positive errors mean delayed responses with respect to target arrival, and negative values mean that they were a little bit anticipating their responses. And I'm plotting here the error as a function of previous trial speed and current trial speed. So I'll ask you to focus for a second here only for the current trial speed at 24 degrees per second to show you that we replicated the results from McKean and collaborators, where previous trial speeds that were slower than the current one led to delayed responses, whereas faster previous trial speeds led to anticipated responses. And this pattern happens across all current trial speeds we tried here. So you can see this decrease in temporal error, or a decrease in bluish, dark blue to light blue here on the 28 degrees per second, or here for 20 degrees per second current speed, a decrease from light red to a dark red. So it seems that serial dependence was present here in our task. And to quantify this effect, we performed a multiple linear regression with temporal errors as our dependent variable, and with previous trial speed and current trial speeds as our regressors. And what we find here is that our beta values are significantly different than zero, showing that we have zero dependence. Negative values here mean that the greater the speed of the target on the previous trial, more participants tended to anticipate, in line with this idea that they were attracted toward previous trial speed. And here, most importantly, we have the stimulation conditions and comparing them to vertex, our control condition. And first, let's look here at the dorsal premotor cortex. When we compare this one, and when we look to vertex or no TMS, it seems that that gradient coming from a higher value to a lower value on the right side of the raster plot, it's somehow decreased. The figure is a little bit more lighter than this one on the left upper corner. And when we compare dorsal premotor cortex parameter values with vertex, we find a decrease in serial dependence. So this is very interesting, and first thing to note here is that this corroborates the idea that the same regions that participate in processing information about a task or stimulus might also participate in keeping information from one trial to the next. And when we stimulate with TMS, we decrease this effect. But does that mean that serial dependence relies on motor behavior or a later post-perceptual process? We don't think we can conclude that with our data. First, because dorsal-premotor cortex also has been associated with, for instance, time perception, motion perception. So it could be also that this region is storing some other information. So we are not sure the nature of information that is being stored in dorsal-premotor cortex. Now, when we compare human V5 with vertex, we don't find evidence for a difference between these two conditions. Does that mean that serial dependence does not rely on visual cortex or visual areas? We also don't think we can say that with our data for many reasons. First, because V5 is not the only motion sensitive region in the visual cortex. V3, V6, some regions in IPS are also motion sensitive, and information could be relayed to these regions, and these should be the places that memory is stored from one try to the next about target speed. Also, it could also be the case that both V5s have some information from one try to the next, because the stimulus spans the whole visual field. And when we stimulate just the right side of the brain, the left side might be able to hold the information from one try to the next and then leading to the bias. So we think it's still, these are still open questions to be resolved. Another interesting thing we think we can interpret from our data is that serial dependence may rely on memory trace precision. And what do I mean by precision here? Coming from this idea of a population coding perspective, where many neurons code for the information about the task being performed or perception being carried out, where some neurons are more sensitive, some other neurons are less sensitive, and all of them somehow code the information. And here, using this idea of a probability distribution representation. And with a wider distribution, you would have less precise memory trace. With a narrower distribution, you would have a higher precision memory trace. And what we think we are doing with the TMS here is that we are stimulating many neurons at the same time, facilitating many neurons at the same time, thus decreasing the precision, because now we have more neurons coding for some information. So we think that TMS might be flattening this distributed representation of the memory trace. And also coming from this perspective that the information from one try to the next are optimally combined based on their reliability or precision, when we flatten this distribution, we would give less weight to the current percept, leading to a lower bias. Now, this interpretation, however, is at odds with recent studies that point out that serial dependence relies only on current trial precision. And this conclusion comes from studies that have manipulated both the precision of current and previous trial stimulus, and have shown that only the precision of the current one leads to changes in the serial bias. But one way I think we can reconcile these differences in results is that maybe the memory trace that leads to serial dependence, its precision, is not directly related to the stimulus precision that was presented on the previous trial. And this would explain why applying TMS would decrease the memory trace precision and lead to decrease to serial dependence, and why manipulating the previous trial precision does not lead to such changes. Cool. So one of the things I like the most about this study I just presented you is that it left so many open questions on the surface. And here I'm highlighting two of them. One that we tried to address in a study recently published, and another one that is an ongoing work. So we are interested in understanding how long does this memory trace that leads to serial dependence last, and what features are stored in short-term memory. So what is the nature of such information that is being stored from one try to the next? And when we talk about the memory trace that leads to serial dependence, we are still trying to find out what is the mechanism responsible for keeping this information from one try to the next. And there is a lot of work in the working memory literature trying to describe what are the properties of such memory traces for longer periods, for short periods of time, but as a function of time. And computational and experimental evidence have described what might happen to the memory trace as time evolves. And here I'm going to talk about two of such properties that I think we can probe using serial dependence experiments. One of them is decay, and the other one is diffusion. Decay is this idea that if the memory trace is represented by a population of neurons over time, as time progresses, those neurons coding more strongly for the memorandum would decrease their intensity in spike in activity or in synaptic weight over time, and this activity would be more distributed across the network. So a decay or an increase in decrease in memory precision or distuning of the memory trace. On the other hand, diffusion is a property that instead of decaying the memory trace stays with the same levels of activity with a bump similar across time. But this bump, the center of the bump, it diffuses, it jumps or wiggles around over time. And this would explain why participants would tend to respond with more variability for longer delay periods. So these are two possible mechanisms for that would affect the memory trace over time if we manipulate this variable in a serial dependence experiment. So this is what we were interested to test here. So what would happen to the serial dependence effect if with the passage of time, or also what would happen to the memory trace that leads to serial dependence as time between trials increases. And this is work spearheaded here by Cristiano Villacchi, together with Esaú Sirius and André Cravo from the Federal University of ABC. And here we were interested to test the limits of serial dependence without intervening trials. So much of the studies that have talked about the length or the duration of serial dependence, or how long it lasts, used intervening trials. So it would talk about the effect of serial dependence for the N-2 or N-3 trials, but did not have tested how long it would last without intervening trials. But one study by Daniel Bliss, where they showed in a working memory task that after 10 seconds, around 10 seconds, serial dependence would vanish. But here we have a task that does not require participants to explicitly keep information during the inter-trial interval, and we wanted to check what would happen to participants' behavior. And so we had participants perform the same interceptive task I showed you on the TMS study, but this time we manipulated on separate blocks of trials the inter-trial interval. So they have 100 milliseconds, 1 second, 2 seconds, 4 seconds, and 8 seconds inter-trial intervals in separate blocks. Before I show you the experimental results, I'm going to show you some ideas we had and simulated in a Bayesian ideal observer model of what we would expect from our participants' behavior if their memory trace was either decaying or diffusing over time, and if those information would optimally combined based on their reliability or on their precision. So this idea, here the decay would be represented by a decrease in memory precision, so a broadening, flattening of this distribution curve representing the memory trace, for representing decay. And for diffusion, we would change the center of the memory trace, like it's showing here. And as time progresses, we would expect more trials, a more broad position for this memory trace, for the center of the memory trace when diffusion occurs, or if diffusion occurs. And here are the simulations of the model. And what we observe is that for a model that just presents memory decay, we see a decrease in participants or in the simulations error variability. So we would expect less variability in participants' responses as a function of an increase in inter-trial interval. And at the same time, we would expect a decrease in serial dependence over time as the memory trace would be larger, would be broadening. On the other hand, if the memory trace just diffuses over time, there's no change in their decay, we would expect here an increase in participants' response error, in their variability, and we would expect no change in serodependence, as the average position of the bump would be always in the same place, despite this variability over trials. And what do we find in our results? First, I just want to show that we found the same effect we found on the TMS study, the same serodependence effect. And interestingly here, when we look at these raster plots, it doesn't seem that serodependence decreases much. These are look very much the same. And sorry here, because the colors are changed. So dark red codes for positive values and blue for negative values. This was an internal dispute about what would be the most intuitive ways to present negative and positive values, and I lost. But that's okay. Now I can present these results without confusing what is negative and positive. And when we quantify this effect of serodependence using, again, multiple linear regression, the regressor for the previous trial speed, it starts roughly at the same point as we showed in the previous study, a little bit higher actually. And once we increase a little bit the intertrial interval, serodependence decreases from 0.1 to 1 second and then stabilizes over time. So it seems that there is some decay from this moment to this moment, from 100 milliseconds to 1 second, but then the memory decay stops, occurring. So precision of the memory trace might not be decreasing over time. And when we look at participants' response variability over time, it seems that participants' variability doesn't change at all over intertrial intervals, which is interesting and not compatible with the idea that the memory traces diffuses over time. And although there's this paper by Daniel Blees and collaborators showing that serodependence for a working memory task for position information, it vanishes over 10 seconds. There's this recent paper by Mauro Manassi and David Whitney, where they find a curve quite similar to ours. So serodependence starts at a higher level here without interstimulus interval, and then it suddenly decreases, and then it seems to stabilize over time, really similar to our curve here. But Mauro and David push it a little bit forward and are now current world record holders for longest time for eliciting serodependence, 15 seconds. So we think this might be, we should also look into other tasks. Here they use a phase perception task, we use an interceptive task. Maybe it would differ for other tasks if this memory trace would last that longer. So with these results, we think that the serodependence, the memory trace, the serodependence relies on a stable memory trace, and that these results are somewhat incompatible with models that predict an increase in diffusion and decay of this memory trace. So these models that are used to explain working memory based on spiking activity usually predict that the longer the delay, the more there will be diffusion in the memory trace, the position of the center of the bump, and higher variability. Also, there should be some decay in the memory trace. So what is this mechanism behind this memory trace? So there are some suggestions in the literatures here by Kilpatrick, Barbosa, and Stein, suggesting that it might depend on short-term synaptic plasticity, but a form of synaptic plasticity that can last at least 8 or 15 seconds. But I don't want to throw out the idea that the memory trace might still depend on spiking activity or of spiking neural networks, because there's this idea, there's these results that different biophysical network models, they can yield opposing predictions on how the excitation and inhibition ratio affect memory diffusion. So depending on how you wire the neurons in the models and how you traverse across the parameter values of these models, you can decrease diffusion. So we might be able to play with these models and find some model that even with spiking neural networks, there shouldn't be diffusion depending on the parameter estimates. I think it's something interesting to investigate further in the future. So now coming back to the other question we had when we finished the TMS study, in this work spearheaded by Esaú, and together again with André, which is part of Esaú's doctoral thesis. We are interested in understanding what is the information stored in short-term memory that leads to serodependence in interceptive actions. And this comes from the result that stimulating dorsopulmonal cortex does not resolve if it is a motor bias. It could be other information stored there. And we haven't stimulated the whole brain for that matter. So there may be other informations that are task relevant, as previous studies in serodependence have shown the information that should be task relevant for the serodependence to appear. And interceptive actions have more than one of such features that are task relevant. Could it be time, the time it takes for the target to travel across the screen? Could it be the speed of the target, the distance, the motor response? So with this part of Esaú's project, we have two goals. One is a more specific one, to investigate which information bias interceptive actions. And the other one is a more broad goal, a more general goal, is to push forward this idea that the memory trace might be stored throughout the brain, across the brain in different regions. And we want to test this distributed and concomitant nature of memory that leads to serodependence. And this is the first study we designed for this project, and we have just finished analyzing some of the data that we collected. We finished collecting data, and so they are fresh from the oven. And the idea of this experiment was that if we fixate one of the features that are task relevant, what happens to serodependence? Is still present after we fixate one of these features? So what are we fixing? We have different blocks, separate blocks, where participants perform this same interceptive task that I showed you. Now with either speed, time, or distance, with not varying over time. So when speed is fixed, we vary time and distance that the target should travel. And these are completely correlated. And the same thing goes when we fix time and distance. And just as a reference, the first experiments we did, we always had distance fixed. So speed and time were always completely correlated. And what could happen in this experiment? We thought that by fixing one of these features, bias could disappear. And if it did, we would be able to say that the feature we were fixating was essential for driving the bias. On the other hand, if the bias persists, it would indicate that the feature is not the only one driving the bias. We wouldn't be able to say that that feature was not relevant to generate the bias. So this is the first experiment, and I haven't had much time to delve into these results. We just discussed them on Friday, but here is what we have. So here on the lines, we have the conditions. So here is when the distance is fixed, when time is fixed, and when speed was fixed. And the columns here, they represent different regressors that we did just for completeness, because the regressors are completely correlated. So we did separate multiple regressions with, for instance, when space was fixed, speed and time as the regressors in separate regressions. And as expected, they are the same, only of course with opposing values here for time and speed, because they are linear, they inversely correlated. So what we find here is that we don't see serodependence disappearing when we fixate either of these features. So serodependence is still present in all of our conditions. So it doesn't seem that any of these features is the only one driving the effect. But we see some differences across these conditions, and we are still trying to understand those. But it seems that when space is fixed, we have the higher influence here when we normalize the regressors to have on the same scale. And when either time or speed are fixed, the serial dependence decreases a little bit. So it seems that distance or space has a smaller contribution than speed and time, but we are trying to figure out also how to interpret this data. And also this experiment cannot rule out other possibilities. So we are now designing a second experiment where we want to probe memory interference. So if we have this idea that the same regions that process the information are also responsible for keeping information during the intertrial interval. If we perform a secondary task that needs the same resources that are being used to keep this information from one trial to the next, we should interfere with these memory traces. So we are designing tasks that would tap on processing information about speed or time or motor responses in between trials, and see if we can change the serial effect, or even if the task we are performing can affect the secondary task. But we are still designing this experiment. Maybe someday I can come back here and talk to you about it. So to sum up, serial dependence is a bias on the current behavior that is driven by previous stimulus or responses. And we have this working hypothesis that the memory trace that leads to serial dependence is stored in the same regions that encode and process information about the task. And I showed you some evidence about it for the dorsopulmonal cortex in an interceptive task. And we also want to push forward this idea and still test it, that it might be distributed across the processing hierarchy. In other words, more than one region might be storing information that would ultimately lead to this combined bias. And that the memory trace seems to be stable over time if we don't have any intervening or interfering tasks or trials. And it would be interesting to understand a little bit more about the mechanism of this stable memory trace. So I'd like to once again thank Andreas, Chris, Esaú, and André for working together with me in these projects. The institutions that allowed us to perform these experiments, the University of Tübingen, the Max Planck Institute for Biological Cybernetics, the Federal University of ABC. I know it might sound funny, but ABC here stands for the cities where the campi of the university are distributed at. So Santo André, São Bernardo, São Caetano, that's why the ABC, where André is a professor, Esaú is a PhD student, and Chris is an undergraduate student. Also my current institution, Instituto Israelita Albert Einstein, the Brain Institute there. Also our funding that provided funding for Esaú, Andreas, me, and André, so CNPq, FAPESP, CAPES. And thank you very much, Mike, once again for inviting me for talking to you and chatting about these projects and your attention. And I'd be glad to talk to you more about these projects now. And if you want to reach out, this is my email and my Twitter handle. Thank you very much.
Thank you. Thank you so much, Raimundo. And I think we are super, super on time. So I'll just keep the recording going. You can obviously trim it afterwards if you want to. Yeah, I would like to invite everyone who's still there to unmute yourself, turn on your cameras, and ask any questions that you may have.
Should I just unshare my screen, or should I keep it sharing? Unshare?
Yeah, I would unshare it.
Okay.
That way I think we have more space for the people. Okay. The obvious suspect. Hello. Yeah, I'm sure you guys have a lot to exchange about.
Hello. Sorry, my computer is very slow, so it takes like 5 seconds to have the thing to appear. I have a question. Oh, first of all, very nice talk. I really liked it. I have a question about the special tuning of serial dependence. So I was wondering if serial dependence is so linked to the task. How do you reconcile the fact that serial dependence depends on the relative location between current and previous trial? Because in that case, the task is exactly the same, right?
Mm-hmm. Yeah. Could it be the case that it would rely on regions that also have some spatiotopic or retinotopic coding? Because I wouldn't say they are mutually exclusive, right? If you have this dependence on space, if the stimuli are close in space, sorry, you're talking about the yeah, I'm not sure if we are talking about this the tuning of the zero dependence effect, that it only close enough stimuli are. You're talking about this one, okay?
Yes.
So my answer was going in another direction. Sorry, I'll start it over. I don't see them. Yeah, once again, I don't see them as mutually exclusive, because it would depend then on how the memory traces are combined. So if the stimuli are similar, they are combined more strongly. If they are dissimilar, they are not going to be combined at all, but not because they are, I don't know how this would impede other regions that are not even retinotopically coded to participate in the task, right? Yeah, not sure I was clear on my answer, but I would say they are not one impeding the other.
Yeah, absolutely. I totally agree. I mean, it's they're not mutually exclusive, and I totally agree that both effects modulate serial dependence. And I just have one comment about the experiment with the delay. I think, if I remember correctly, I also tried something very similar with increasing the delay in between trials to see where serial dependence was going to end without intervening trials. And if I recall correctly, I also still found serial dependence for a delay of 12, 15 seconds, and I had to run another experiment, which was really excruciating for the subjects because it was something around 24 seconds delay, and I found it. This is all to say that I think it is there. I just think it's about testing a larger delay, which I know it creates an incredibly long and painful experiment for the observers. But yeah, this is my experience. I mean, I don't think I have collected too many data on that. I think it was only 6-7 subjects, but I think it would definitely point in that direction.
Yeah, we thought also about doing a follow-up study to see how long it would last. But yeah, it was really, really hard to have participants perform the 8 seconds. Was already really, really long. Like your experiment, the one with the faces, participants don't have to do the task for a long time, right? These are one-shot trials. Then it would be maybe more feasible than performing many tasks as the other experiments I know you have done. And this one, because participants, they perform 255 trials per condition, and for 8 seconds, that's a long time. I just want to say that the world record, it's only official when you have the paper published with the record, right? The 24 seconds doesn't count as a record. You have to submit to Guinness. Yeah, but that's good. Really good. That's really cool to know that it lasts 24 seconds as well. Was that for the?
No. What I'm saying is that for 24 seconds it decayed. So I think for 24 seconds it was even negative.
I understand.
So I found it for 5, 10, 15, 20 started decreasing. It was around 0, and 24 it was even negative.
Cool. I understood it incorrectly. So 20 seconds it started decaying. It's a long time anyway.
Yeah, I agree.
Cool. So the
It
record
was a very long experiment. I remember it very clearly.
for which task was it? For the face or no, or the other one?
Orientation.
Orientation. Okay, so really long experiment. Cool. Thanks for the questions, Mauro, and also for sharing the length of serial dependence. What is out there?
Thanks for the talk.
Thanks.
Yeah, and we have another question in the chat. I don't know if Elena is still, yes, you're still with us. I can read it out loud, or you can unmute yourself, however you want. Says, do you think adding, sorry, I think you just muted.
Oh, no, all good. Well, I'm currently working, I'm a graduate student, and I'm currently working on a serial reaction time task and looking at audio visual contributions. And this question's just popped into my mind, how if we have more redundant information, how that might influence serial dependence. So if we only do these tasks as visual tasks, or doing bimodal tasks such as audio-visual ones, if the targets, for example, are enhanced or augmented by auditory information. If that makes any sense, yeah.
Yeah, I think it's a really cool idea to test how different modalities influence. You mean one behavioral outcome, right? You have
Yes,
one behavioral
exactly.
outcome,
Yes.
but you have two features from different modalities, which is somehow similar to our task here, right? We have one task, one modality, but it has more than one feature that is task relevant.
Yes.
And I think your idea is really cool to test. Like you have two different features from different modalities. You can really separate them. So I think it would be really nice, and see how both affect behavior, right?
Yes, yeah, absolutely. I was, I don't know, my mind's going different places. I thought if we have the targets moving, for example, they could either be augmented by maybe the same tone, or if different speeds are presented, then the targets would have varying frequencies or pitches. Yeah, it's more of a very vague vague description, but it just kind of popped into my head.
Yeah, I have some speculative thoughts on the idea, but I think it's really worth a shot, right? It's interesting. But when you have two modalities, you usually have these how these are going to be combined to generate the final percept.
Yes.
And there's all this discussion that depending on the reliability of either source of information, one is going to be higher weight and then going to drive behavior more than the other. So I don't know how these two would affect serial dependence. So I think there are these two components that have been thought through. So you have serial dependence, the memory trace that might affect behavior from the auditory and visual modalities, but there is also how they are going to be combined based on their reliability.
Yes, that makes total sense. Maybe, yeah, more temporal information stored through the auditory modality, while spatial location would I guess refer to visual information. Yeah, thank you so much.
Thank you for the question. I think it's going to be a nice experiment. Yeah, I would like to see it.
This is your invitation, Elena. Yeah.
Wow.
Okay. Any other questions from the people who are still there? This is your chance. Okay, I guess not. So yeah, if you have any other questions or feedback, obviously feel free to reach out to Raimundo. And I just want to say again, thank you so much for giving an excellent talk. I thought it was super cool. Definitely gave me a lot to think about. And yeah, I look forward to catching up with you again later.
Yeah, it would be nice to come back here once we get more things to present. Yeah, thanks for the invitation again, Mikey. Thanks for the questions. And see you around.
Okay, thank you. Thanks, everyone. Bye.
Thank you very much.
Thanks.